The scientist I chose as my role model is Dr. Jack Gilbert. He currently works at the University of California, San Diego as a professor in the pediatrics and the Scripps Institution of Oceanography. He also is a group leader for microbial ecology at Argonne National Laboratory, along with many more positions. He had always had an interest in science and medicine as a kid and went on to get his PhD from Unilever and Nottingham University and his postdoc at Queen's University in Canada. Dr. Gilbert grew up in the UK, so he eventually had to overcome moving to the United States to work at the University of Chicago in 2010. In his free time, he likes to spend time with his kids outside and he likes to listen to classical music. What I admire most about Dr. Gilbert is his never-ending enthusiasm about his work. He also works a lot with the significance of the microbiome in our everyday lives and how important it is to maintain a healthy microbiome for our health. This is something I am extremely interested in and hope to work with the same topics in the future. I identify with him because of this. Dr. Gilbert works a lot with the microbiome of organisms and their environments. He answers questions that deal with how microbes are transferred throughout everyday activities, how the microbial ecosystem health can be measured in the Chicago River, figuring out a way to alter gut bacteria to improve symptoms of mental illnesses like depression and anxiety, the microbiome of many different species like bats and African wildlife, and many more topics. The most recent publication of his is called Longitudinal Microbiome Composition and Stability Correlate with Increased Weight and Length of Very Low birth weight infants. In this experiment, they tested to see the correlation between the microbiomes of the mother and infants at different ages, and if the type of milk fed to the infant affected the bacteria present in the infant, and if the microbiome of the baby then led to different growth. To identify the bacteria/microbiome present in the subjects, they collected their stool every week. They used PCR to then identify the DNA in the stool and then match that DNA to different bacterial species. The species were then compared with other infants based on the type of milk they got fed, their weight and the height each week, and additionally compared to the species in the mother. As seen in the first figure, there was a correlation between the toddler's microbiome and the mother's, but the infant's was more different. Looking at the second figure, it can be seen that the number of bacterial colonies are most similar in the healthy growth of infants, where when the data spans away from the normal bacterial colonies, more abnormal measurements of the infants were taken. This allows for the conclusion that by the time a child is a toddler, the microbiome is almost identical to that of the mother's, and the microbiome is hugely correlated with the weight and height of infants. Dr. Gilbert has uncovered the important links between the gut microbiome and the brain and is a huge contributor to increased awareness of the importance of the microbiome in childhood and development and in our everyday lives. He co-authored a book called Dirt is Good, which is targeted towards parents who want more information on how to allow for healthy microbiomes in their children and alleviating concerns that they have about being dirty. The next steps in his research include finding ways to use these newly discovered links between bacteria and previously inexplainable phenomena to solve these problems or create new therapies to treat medical conditions.